confusing, don't be afraid to look at that and let me know what's going on. So first things first, you have 3 chapters on this next exam: senses, blood, and cardiovascular. So we covered everything except for that second part of cardiovascular, which I recorded for you last week, actually. So I have it already up for you. I will send you a little bit of information on that in an email here soon, later tonight. But regarding the exam, let's start with senses. What's going to be on there? You are going to have a couple of diagrams, the ear and potentially the eye, that you will have to identify structures of. Okay, so we'll just point to one. You'll tell me what that structure is. They're going to be the big things: the retina, the sclera, the pupil, the auricle, the ear canal, whatever it might be. There's going to be larger structures, but please be aware of those things. Also, parts of the eye, parts of the ear, just understanding what they do. So functionally, these 2 structures are very, very important to us. So please make sure you come in knowing what those are. Okay, so I highlighted those for you. We talked about things like the lacrimal glands. We talked about the sclera. We talked about the iris. We talked about the pupil. We talked about the choroid and the retina and the photoreceptors and the optic disc and the fovea centralis. Okay, so please make sure, ciliary body, make sure you are aware of what those structures are doing. Okay, also with the ear, generally speaking, the ossicles, the auditory canal, the external acoustic meatus, all those parts that we've discussed once again are going to be important to you. Know what your photoreceptors are doing, those rods and cones, and the parts of the eye, once again, that I've already discussed. Okay, make sure you know the sensations of taste. There were 5 of them. Be sure you can locate those. And then generally speaking, how taste works. Remember, smell and taste are both going to be by chemicals that will catch on to those hairs, either up in the brain or also then on the taste buds, that are going to allow then that signal to be sent to the brain. All right, so make sure you're aware of those different things. I've given you some detail on what parts of the eye give you the best vision and which parts do not. Okay, so best vision, remember, is on that fovea centralis. That's 20/20. That's where the lens is going to bend that light to hit. If you have glasses or you don't see 20/20, it's because your lens bends light and it doesn't hit on that fovea centralis. And so you wear glasses to fix that for you. Make sure you know the definitions for emmetropia, hyperopia, and myopia. Okay, those are going to be very, very important to you. Perfect vision, farsightedness, and nearsightedness. When you talk about the ear as well, once again, understand what the parts of the ear are doing in a large scale, like we talked about with the eye. And then your 2 types of equilibrium, static and dynamic. Be aware of what parts are there and what happens with the rotation of the head or the otoliths or whatever structure was involved. Also, I've mentioned to you before, be sure you are aware of the nerves. If I gave you any cranial nerves that those senses are carried by, make sure you know them, okay? All right, so let me just skim this here real quick to make sure we're good. So 2 words that were given to us was smell and taste. Olfaction is going to mean smell, and gustation means taste. So make sure you can determine those anatomical terms there. And then moving on then to blood. When you talk about blood, there's lots of characteristics of blood. I asked you to know, for example, the temperature is typically right around your body temperature, a little bit warmer. You're going to have your pH. Make sure you're aware of the cells that are found there, that they're the majority red blood cells. You also then have that buffy coat. That's going to be the platelets and leukocytes. And then you have the rest plasma. The plasma is mostly water, a little bit of protein, a little bit of electrolytes. So make sure you're aware of those. When you talk about the plasma and those proteins, be aware of the proteins that I gave you in class. So I gave you albumin and I gave you fibrin, and fibrin is going to be the protein that helps you clot, and albumin is going to be the most common. That's going to be your transport protein, also helping with osmotic pressure, which is what's in your notes. When you talk about erythrocytes or red blood cells, remember there's tons of those guys. They have a lifespan of about 120 days. They're biconcave, meaning they do not have a nucleus. And so they don't live all the time. They get recycled in the liver and the spleen, where those guys will be taken back up and will reuse their constituents. When you talk about red blood cells, there was one thing that I do not think I mentioned to you, and I apologize about that, but it was talking about the percentage of red blood cells. And the percentage of red blood cells in the body is known as hematocrit, Hematocrit, hematocrit. So if you see that on the exam, that's what that is. We also talked to you about hemoglobin. Hemoglobin is the protein that carries oxygen. I gave you a couple of numbers there, specifically knowing that every hemoglobin carries 4 oxygen, okay, is very, very, very important to you. When we talk about the oxygen carrying capacity, I gave you a couple of scenarios. I gave you polycythemia and anemia. Anemia, remember, is low carrying ability, and polycythemia is more. So make sure you're aware of this terminology that we're looking at with these different conditions. When you talk about leukocytes, the white blood cells, it's no different. I gave you a couple of conditions: leukopenia, leukocytosis. So make sure you know the differences on those. And then a couple of terms before we get to blood typing. Agglutination and coagulation. I had you guys talk a little bit about when we did that worksheet in class, but agglutination, agglutination, is when an antibody attacks an antigen, okay? So remember, that's what happens if you put the wrong blood into somebody's system, where the antibodies of that one person that you put the blood in, they'll attack the antigens of whatever you stuck in there. That's called agglutination. Coagulation is very different. Coagulation, which you've seen in the notes, it basically means to blood clot. Okay, so that is the process of stopping blood from leaving your body. So if you get a cut, your body is going to undergo coagulation. That's important. That is clotting in essence, but it is not the same as agglutination. So make sure you keep that straight. Hematopoiesis is another term we talked about. That is going to be where we form blood cells. That happens in the red bone marrow, which is at the end of your long bones. When you talk about blood typing, guys, you have to know on this exam, it comes up numerous times, what antigens are present and what antibodies are present and what blood types you can get or receive, okay? So when you look at the blood typing aspect of this exam, it's not going to be tricky, but make sure you either draw it out if you need to or recall that diagram I gave you. When I look at blood clotting, remember, there was one tagline I gave you and I said, if you are a blood type, that's the antigens you have. So if I am blood type A, I have antigen A. That would mean in my plasma, I have antibody B, okay, because those should not match. If they match, I have agglutination, all right? So we don't want that to happen. So make sure you're aware of blood typing. If by chance that's tricky for you still, please contact me. I have a whiteboard here at my house. I can draw that out for you again if I need to. We can chat about it, whatever I need to do. I just want to make sure you understand it. When we talk about the baby that may get that with the Rh factor, remember, that's called hemolytic disease of the newborn. I mentioned that as the only time an Rh will be a factor for you. All right, so the Buffy coat, I already mentioned to you, once again, being part of that whole blood. You have erythrocytes, Buffy coat, and then plasma. Buffy coat, remember, is platelets and leukocytes, okay? Platelets and leukocytes. Platelets are not true cells. They do not have a nucleus. They come from that megakaryocyte that we talked about. They're just pieces of cells. The white blood cells, there's 5 types that I gave you. Some are agranulocytes and some are granulocytes. Way to remember that. Granulocytes are Ben, B-E-N, basophils, eosinophils, and neutrophils. Agranulocytes are the other types, monocytes and lymphocytes. You will want to be aware of what each of those cells respond to. On the exam, I will either just simply ask you what they respond to, or I'll give you a scenario where I'll say, look, Sally has a viral infection. Which blood cell would be elevated? And you would answer lymphocyte, because viruses are going to be detected by lymphocytes, okay? Alrighty. Now, if any of those things are tricky, let me know. Otherwise, we're moving right along here. I also will ask you about mixing blood. So I'll give you a scenario where I say, look, Susie wants to give Sally some blood. Susie's type A, Sally's type B. Why can they not do that? Okay, so you'll have to pick the answer on that. That makes most sense. You're also going to see, moving on to cardiovascular, there's some written questions at the end that I'll explain more about these chapters, but that's it for the multiple choice for blood. When you get to the cardiovascular, you are going to see a heart diagram. You will either have to label heart parts, or you'll have to label the parts for the electrical conduction of the heart. And that is well versed on that online lecture I gave you. You're also going to have to talk about the base and the apex, so the parts of the heart, maybe the layers of the heart as well. So be sure you can tell me the differences there, along with what valves come when. Okay, so when you talk about these valves, I may just say, hey, what's the name of the valve that sits between this ventricle and this atrium? Okay, so you'll have to just tell me the valve at that point in time. The other things you're going to see here are the differences between pulmonary and systemic circulation. You may either have to describe it to me or just tell me which one is. So I'll say, this is the type of circulation between the lungs, and that's going to be, or to and from the lungs, I should say, and that's going to be pulmonary. This is the type of circulation from the heart to the body, and you're going to say systemic. Okay, it's that simple. I may also ask you what type of blood is coming to and from those. Remember, pulmonary takes deoxygenated blood and gives you oxygenated. Systemic takes oxygenated and brings back deoxygenated. So be sure you're aware of the blood types there. All right. And the pacemaker of the heart is the sinoatrial node. When you look at that, once again on that online lecture, make sure you're aware of the parts of that electrical conduction. You'll label that for me. You'll explain the whole pacemaker idea, and then you may have to put those steps in order. So be prepared to do all of those things. When you see things like the sounds of the heart, lub, dub sounds, that's the closing of the valves. LUP sounds is the closing of the AV valves. DUP sounds is the closing of the SL valves. So the AV valves are the tricuspid bicuspid valves. So that's the LUP sound. The DUP sound is going to be the aortic and pulmonary valves when they close. So keep that in mind. There's also a lot of terminology in that chapter that I gave you online, and I hope that you have taken the time to listen and get through that, because if you don't understand those terms, this test is going to be tricky for you. So systole and diastole are two words that come up a lot in that chapter. Systole is going to mean contraction. Diastole is going to mean relaxation. So this can happen in either atria or ventricles. If the atria are undergoing diastole, that means they're filling, they're relaxed. If the ventricles are undergoing diastole, that means they're relaxed and they're filling. So, and the same works with systole. You'll have to understand heart rate, the heart rate of an individual, and things like stroke volume and cardiac output, and how those three are related. You're going to see the equation on this exam. So on the lecture, once again, I gave you that cardiac output, meaning how much blood you're getting in a minute, equals stroke volume times heart rate. So stroke volume is how much blood is leaving the ventricle every beat, and then I'm taking that times the heart rate. So heart rate times how much blood is exiting per beat gives me how much blood I get in a minute. That would make sense. So make sure you can look at that equation and make sense of it for me and define the parts. Okay, know what cardiac output means, know what stroke volume means, and know what the number is for the average heart rate. Okay. You also need to know the difference between artery and vein and what they look like. Okay, so arteries are highly muscular, veins not so much. All right, those look very different. They also, veins have valves that keep that blood flowing because they're under a lower pressure than the arteries are. Arteries are under a higher pressure, so they're thicker, and they move that blood a lot faster than veins do. You also need to know the difference between vasodilation and vasoconstriction. So you have these vessels, okay? And when you talk about these vessels, they can sometimes get bigger or they can get smaller, okay, depending on what's happening in the body. I want you to know definition-wise what is occurring here. So if I have a vessel and it gets larger, that means it dilates. It's called vasodilation. If that vessel dilates, my pressure is going to slow down, okay, because I got more stuff flowing through there. It's going to slow down. If I vasoconstrict, getting smaller now, if I vasoconstrict, at that point, my pressure goes up, okay? Things go faster. This would make sense. Think about, playing with a hose when you were a kid or, outside, even it's been kind of hot around. If you have that hose open, just open, water's going to be just pouring out of it, right? But if you stick your thumb over that hose and decrease that diameter, what happens to the pressure? It goes ding, and you're able to hit your brother or your sister, right, or your family member with the hose. All right, so you kind of get the idea there that vasodilation, lower pressure, okay? Vasoconstriction, higher pressure of blood inside of that system. So if I ask you, you need to speed up the blood inside of your body, what happens? Vasoconstriction. You need to slow down the blood in your system. Your heart rate is too high. You've got too high blood pressure. What happens? Vasodilation. Okay. Now, there's a lot of teaching that goes along with that is really complex that we haven't talked about yet. So that's not always the case, but the teachable point is just to say, if you constrict something, it goes faster. If you dilate it, it goes slower. Okay. And we'll take that and we'll expand on that a little bit in a couple of chapters. All right, you have to know your blood pressures. I give you a bunch of those. I give you an average 120/80. I give you a hypertension and a hypotension value. I also give you tachycardia and bradycardia. So be sure you're aware of all of those terms, along with what vessels carry what blood. Okay, so I'm going to focus on large vessels, but you need to have a pretty good idea specifically on the vessels off the heart, whether that blood is oxygenated or deoxygenated. Okay, now you colored that. I mentioned to you, you've seen pictures all throughout what we've done, blue and red. Make sure you know, is the aorta oxygenated or deoxygenated? Is the vena cava oxygenated, deoxygenated? Is, different parts of this heart. Make sure you know, are the pulmonary arteries oxygenated or deoxygenated? Are the pulmonary veins oxygenated or deoxygenated? Those are the four biggest ones. Make sure you also know coronary sinus and coronary arteries. Are they oxygenated or deoxygenated? Okay, and you should be good. Also know what affects your heart rate. So what would speed it up? What would slow it down? Okay, there are some obvious answers in the notes that I gave you online. Things like adrenaline or epinephrine increase it, caffeine increases it, stress increases it. You're only going to see a few decreases. Parasympathetics will decrease your heart rate. So keep in mind there's going to be some slowing of things overall. Make sure you know what increases and what decreases. Also, the layers of the arteries and veins, I gave you that as well. The tunic, they're called tunics. You have a tunica externa, a tunica media, and a tunica interna. Make sure you can put those in order. And then the layers of the heart, okay? Don't forget the layers of the heart. That comes early in the chapter. All right, so other things to know for things like finishing up this exam, which I think I've given you most of this already. Good kind of thinking question is why the left side of the heart is thicker than the right side. Remember, it's about flow, where it's going. You're going pulmonary on the right side, so it's on the same level, so it's not going to need as much pressure resistance as the left side would. And then also be able to explain to me why you could or could not give somebody a specific blood transfusion. So once again, this will be a written question. You'll have to explain it to me, and then I'll check that for you and make sure you're on the right line. So if you have Susie who has a certain blood, she wants to give to Sally who has another blood, tell me yes or no why this could or could not happen, okay? I'm giving you some examples of that. We've talked about that all the way through. Typically, it's because of antigens and antibodies, but to get full credit on this exam, because I've taught it to you already, you have to be specific. You cannot just say antigen and antibody doesn't mix. Well, which antigen and which antibody don't mix, okay? And be clear with me on that. The last thing you're going to do for me is put all the flow of the heart in order, okay, for blood flow. you'll have, I think I actually give you the part, I give you the parts, you just have to put them in order for me, okay? So you'll write that in on the exam. As I mentioned to you before, okay, this is going to be a timed exam. There's right around 60 questions, multiple choice, and then a couple of written for you. So keep in mind, time is of the essence. If it takes you a long time to type, you know, that written question is like 10 points. So you don't want to save that to the end because you were looking at your notes the whole time to get everything else together. So you're going to want to have an idea of these concepts. So please listen to this part of it and come in prepared when you do sit down to take it. That you can make it happen, okay, in the time allotted. I have not decided how much time yet to give you. I'm leaning towards giving you what you would have in class because it's written for that. In fact, it's a little shorter than the actual written exam because I made a few of the written questions multiple choice since it's online. It's a little bit easier for grading purposes. The other thing I will tell you is that once you start this exam and you open it, you have to finish it. So you cannot open it on a Tuesday and go back to it Wednesday morning. It's not going to work. It'll kick you out. You'll be done. You'll get a 0. You'll get the points you got. Okay, so make sure you have the time to sit down and complete this exam once you do it. So that's really all I have for now. I will be in communication with you. If we need to have Zoom meetings, we totally can. I was just nervous tonight to do that and then not have enough time to go through the actual stuff I need to talk about. So email me if you guys need anything at all. I am accessible.